Again, thank the morning speakers for very good talks, staying on time, and it's a pleasure for me to introduce my colleague, Dr. Aiku Duren, to introduce the keynote.
Welcome to the keynote speaker for CCF this year. It's a great pleasure to have Dr. Patrick Grohar. He's an old friend and colleague. Patrick got his undergrad degree from Villanova. Then he received his Ph.D. and M.D. from Wayne State. Then he came close by to Baltimore to do his residency in pediatrics. And following that, he did his Heme-Onc fellowship in a program. There was a joint program between Hopkins and NIH, and during that time, he started working on Ewing sarcoma and start doing some screening works. After the fellowship, he started his faculty position at Vanderbilt, and then he moved to Van Andel Research Institute in Michigan as an associate professor. And finally, he moved to Philadelphia to Children's Hospital of Philadelphia CHOPS, where he's currently an associate professor. He also has an endowed chair named after Kelly and Chad Panchart. He also has a faculty appointment with the School of Medicine at UPenn, and he is the director of the Translational Research program at CHOP. He has leadership roles at Children's Oncology Group. We really benefit greatly from his leadership in the biology program for osteosarcoma and Ewing sarcoma. And Patrick is a true translational scientist. Like most of us, he does the bench to bedside work, so he does screening work and discovers new molecules that can be used in the clinic. But unique for his research, he also does the opposite. He closely studies the patients and how they respond to different drugs, and then finds out why, what was the mechanism, how those drugs were working or not working for given patients, and uses that information to develop new mechanisms and new drugs. So his work on mitromycin and trabectedin resulted in multiple clinical trials involving Ewing sarcoma patients. So it's a great pleasure to have him today giving us our talk here. Thank you, Patrick.
Well, thank you very much for the introduction. It's really great to be back here. It's such an honor to be among so many great colleagues and friends and people that I hold in such high regard at all of these institutions. When I was here, we didn't have this, and so I think that this is really an amazing symposium, and you guys are very lucky to have that now. All right. So Aiku gave me a nice introduction of what I'll be talking about, and this is sort of integrating the bench with the bedside to try to advance therapeutics for Ewing sarcoma. These are my disclosures. All right, so when I think about Ewing sarcoma, I sort of think of two groups of patients. Patients with small localized tumors who do very well, 87% 5-year EFS in the most recent COG study, and then patients with metastatic and relapse disease who don't do quite as well. Now, my lab has primarily focused on the relapsed patient population, and that's because there's a great clinical need. So this is a meta-analysis of the last 62 published phase 2 studies in Ewing sarcoma, representing over 800 patients, published by Natalie Gaspar. And you can see the response rate on phase 2 trials with either a Ewing's cohort or Ewing's dedicated trials was only 10%. That is, only 10% of those patients had objective responses, being either CRs or PRs. And only 18% of these studies were positive, and they were positive with pretty dismal results. So a 15% objective response rate is a positive study, or a 4.5-month progression-free survival. And so there's clearly a need to understand why this is, okay? And so the question that I've focused my lab on is, why are we failing to move agents effectively from discovery campaigns or new molecules that are percolating down to pediatric oncology into these patients? And why do we continue to have studies like the last 5 phase 2 studies that I show here on the right in the COG with Kaplan-Meier curves that look like that? Are we using the wrong drug? Is it the wrong schedule? Is it the wrong target? Or is it something about this patient population? Is there something different about this group of patients that are relapsed? And maybe this shouldn't be the patient population that we're testing new therapies in. I think it's a complicated answer, and I think it's going to take many years for us as a community to sort out. But in many ways, I've sort of built my lab around this basic concept as I could introduce there. And the idea is that, if we really focus on the process of going from discovery to the clinic, that might be a way where we can, move the needle forward. And the idea is that this is sort of a circular thing. So you identify a new molecule, you move it to patients, in a trial where you obtain the correlative biology that's necessary to figure out what happened and what went wrong, and then you can sort of go back to the bench and identify sort of mechanisms of resistance, target those, second-generation inhibitors, second-generation combinations, and the like. But if you notice, at the center of that sort of circular process is mechanism. So we need a better understanding of mechanisms. So mechanisms of drug as a biomarker of activity, mechanisms of a resistance as a biomarker of a bad tumor, okay? And so, in many ways, this is where we focus, is on drug mechanisms, with the idea that we can identify new agents, second-generation analogs, and move compounds that we have that are active into other histologies and even other tumor types, which we've published on. But most importantly, we think it's absolutely critical for understanding how to give a drug to a patient, right? So rather than designing trials based on like a serum level, why not design a trial based on what concentration or drug exposure you need to achieve a mechanism? Okay, and if you have that, then you could probably move compounds more effectively to the mouse, which is, in my opinion, the gold standard, tumor regressions in a mouse, and then therefore into patients by designing trials that are mechanistically focused with correlative biology and that are pharmacokinetically and pharmacodynamically driven. All right, so like Jeff and others, we've spent the last 15 years focused on, trying to figure out ways to drug this undruggable target EWS fly. And so this, again, in this group probably doesn't need any introduction, but it's absolutely the driver mutation of Ewing sarcoma. It occurs in a genetic background characterized by few other mutations, cooperative mutations, and those that are there actually cooperate with the fusion protein, has been shown for STAG2. And the dependence on this target has been known for more than 25 years with multiple independent studies, okay? So antisense DNA, siRNA, CRISPR-Cas9, you silence EWS fly, the cells don't grow. And so this is absolutely the definition of a good drug target. And so you'd reason that if you were able to inhibit this target in patients, that responses should not be subtle. And I put this slide up just to capture how, a few of us, including Jeff Turetsky and Kim Stegmeyer, took this on years ago. It's been, almost 15 years now where we've all been working on this idea. And in the last couple years, it's been sort of exciting to see these trials materialize in patients. And I think we are actually learning things that'll be transformative for the disease. But I'm going to tell the story about sort of this circular bench to bedside and sort of leave you with where we're headed. With a molecule called trabectedin. Okay, so as Aykut said, this is a molecule that at the time we were doing screening efforts similar to Jeff, but we originally became interested in this molecule based on clinical observations. And so the clinical observation was that in the background of all these negative studies, that patients with sarcoma were responding to trabectedin. Okay, and the patients that were responding tended to favor those with translocation-positive sarcomas, and the big winner, being more of an adult tumor, was a tumor called myxoid liposarcoma, which is driven by the fused CHOP oncogene. Subsequent to these observations, Maurizio D'Incalci in Italy reported that this drug actually poisons the fused CHOP transcription factor. And so this got us wondering, FUSE and EWSR1 are family members. And then in the phase 1 study of Ewing sarcoma, there was a complete response of a patient to single agent trabectedin. It was a phase 1 across all histologies, obviously. And there was a complete response of a metastatic patient to trabectedin. And so we asked the question, well, we designed all these assays to measure EWS fly activity. Maybe it inhibits the fusion protein, because it's hard to imagine a widely metastatic patient responding to something that didn't target the driver oncogene. And so we published a paper more than a decade ago where we showed that, in fact, the drug does reverse the transcriptional activity of EWS fly, and we showed this using a number of assays, mRNA, protein level, genome-wide. But actually my favorite piece of data was that we could take EWS fly, clone it into another cell type, activate a reporter, and then completely rescue that activation with trabectedin. So we were quite confident that this drug reversed the activity of EWS fly. The problem was shortly after I started my lab, there was a paper that came out that trabectedin doesn't work in Ewing sarcoma. So this was a negative study. It's like not the ideal way to start your lab, right? Like you come out, this is the drug I'm going to work on, and negative study. So, but whatever, we forged on. But anyway, there was 11 patients, and there was one patient with stable disease. And in this study, the drug was given as a 24-hour infusion, which was in fact different than what was given to the patient that had the complete response. And having done the experiments myself, I totally believe the data that we inhibited EWS fly. And so in many ways, this led to the advent of the approach. The idea was, well, something has to be wrong, and we recognized the PK probably wasn't right, and reasoned that what we really need then to make this drug work is an understanding of how it inhibits the fusion protein. And if we had that, then we could probably administer it more effectively, and we also would probably benefit from a potentiator. And so, over the next decade, we published a series of papers, and we showed that the drug works by redistributing EWS fly in the nucleus to the nucleolus. So I'll just summarize that data here. So that's just a single nucleus of Ewing sarcoma cells in culture treated with trabectedin. And you see sort of in green EWS fly diffusely throughout the nucleus, and with drug treatment, it sort of relocalizes to the nucleolus. And what this does is trigger an epigenetic switch, which leads to histone post-translational modifications and silencing mechanisms at EWS fly target genes. So I'm showing on the bottom there chip data of H3K9 trimethylation, and you can see that it piles up at a really well-established EWS fly target, NR-OB1, right at the GGA microsatellite. And if we sort of took where these histone PTMs fell in the genome and subsetted them on microsatellites associated with EWS fly targets, almost all of them were marked with either H3K27 trimethylation or H3K9 trimethylation with trabectedin or lurbinectedin exposure. And so for the purpose of this talk, the important thing to realize was this absolutely was exposure dependent. So exposure equals a concentration of a drug times time. So if you give a drug over a long period of time, you get actually a higher drug exposure, but at a lower concentration. You give it rapidly, it goes high and then clears fast, okay? And so we did some simple experiments where we compared identical drug exposures, comparing those two variables, a low continuous exposure to a short intermittent exposure, and showed quite convincingly that only if you exceeded a threshold concentration of drug would you inhibit EWS fly. And so that's what the Western blot on your left shows, comparing lane 3 and lane 4. Those are identical exposures of the drug. So it's 1 nanomolar of the drug for 18 hours versus 18 nanomolar for 1 hour. Wash out the drug, let the cells sit for 17 hours, and collect the cells and make lysate. And you can see only in excess of a Cmax in lane 3 do you see suppression of targets, WRN, EZH2, and NR-OB1. And similar effects could be demonstrated on viability. So all those curves, that's a graph of cell proliferation over time. And all those curves have the identical drug exposure. And only once you exceeded a threshold did you see sustained effects on proliferation. And in fact, if you could clear 20 nanomolar drug, as little as 6 minutes of exposure would have effects that lasted 3 or 4 days. All right, so we recognize that, well, okay, so in the phase 1 study where we had a response, that was given as a 3-hour infusion, so that should have had a higher threshold. Maybe that explains it. But if we gave the drug as a 1-hour infusion, we should far exceed the threshold that was necessary, and this was based on published PK data from the early 2000s from Europe. And then in the effort to search for a potentiator, we looked for changes in the EWS fly transcriptome that might sensitize to a second agent, identified suppression of a DNA damage response pathway that would sensitize to the DNA damaging effects of irinotecan, and subsequently showed that it's a topo-1 inhibitor, so it has effects on transcription as well, that the irinotecan potentiates the suppression of EWS fly. So it's a really nice mechanism of drug synergy. Drug A sensitizing to drug B, and drug B feeding back and amplifying and sustaining the effects of drug A. And then lastly, one of the things that was really encouraging for us is anytime we gave this combination to xenograft, we saw really rather stunning and striking tumor regressions. And I'm showing data from our original manuscript in 2 different cell lines. You can see on the left, those are, on your left, those are individual mice in a cohort at a given day where you can see the effect of trabectedin, irinotecan, and then the virtual elimination of all the tumors in the combination, this prolonged survival. And then in the right is TC32. Those are spaghetti plots. I'm not showing the control cohort. We've all seen those plots. They grow like crazy. And you can see that with just where the arrows indicate the doses, Each of those individual lines is the growth of an individual tumor in an individual mouse. You can see that every tumor in every mouse goes away. And similarly with lurbinectin irinotecan on the bottom, that's in a PDX model that we had established at the Van Andel when I was there. And then in 2 additional cell lines, we haven't published this. These are actually type 3 fusions. You can also see responses that are quite profound across all tumors. So if this is the gold standard, this seems like an approach that would be worth testing in patients. And so this is the study that many people here at the NCI have enrolled patients on. It's a phase 1/2 study of trabectedin in combination with low-dose irinotecan with an imaging-based biomarker. These are the co-investigators and the sites where we're open, but I do want to just highlight, quickly, how as a laboratory scientist, primarily a laboratory scientist, when you have such an esteemed group of colleagues that sort of rally behind your ideas and your data, it's really about as good as it gets, really. And so that since we're in DC, the NCI has really vigorously stood behind this trial, enrolled a number of patients, and so in particular I want to recognize John Glad and Mary Frances Wedekind, who've been really tremendous colleagues. But we've also worked with PK at the NCI and the imaging, which I'm not going to really talk about. And so this study was called SARC-37. We've now completed the phase 1. I reported at ASCO a couple weeks ago, so I'm going to tell you about the results. All right, so we enrolled a total of 20 patients. It was a heavily pretreated cohort. So if you look at our accrual by dose level, we had a median of 4 prior lines of therapy. So getting to my initial question about are we testing new agents or new combinations in the wrong patient population, that's a lot of therapy, right? So 4 prior lines with a range of 2 to 9. A patient who had 9 prior salvage regimens went onto our study. Almost all the patients, so over half the patients had prior irinotecan. Many had progressed on irinotecan before they got the irinotecan in our study, with over 60%. And nearly all the patients were metastatic. Our patients did span the age range. We enrolled at the University of Michigan, which was our medical oncology site, and so the age range was from 10 to 59 who enrolled on our studies, and there were far more males than females, almost 3 to 1. The toxicities, so we had a fair bit of toxicity in the study. Again, it was a heavily pretreated population. We had 2 grade 5 events that were deemed likely unrelated, although one was a pretty sick patient, so it's hard to really know in that setting, and might have been a clot. But in general, the drug seemed to be better tolerated as a 1-hour infusion than as a 24-hour infusion, which if you think about what I originally talked about the pharmacology being less total drug, it would make sense that it would be better tolerated. We saw no grade 4 liver toxicity, which is sort of the signature of this drug. But a fair amount of myelosuppression. It's still chemo. It's repurposed chemo, focusing on a specific mutation. So there's definitely room for improvement from that. So we saw a fair amount of myelosuppression. We saw no diarrhea at all, actually, at these doses of irinotecan, which was good to see. And in general, the most common side effect was just a feeling of fatigue, and quite profound fatigue, actually. So we had one patient that came off study because they actually couldn't get out of bed. And that was in an adult patient. So that also seems to sort of track with age. So definitely room for improvement in the toxicity arena. And this is the waterfall plots of responses on the trial. So you can see we had a number of patients who had rather profound tumor regressions. Okay, so for those of you who aren't familiar with looking at waterfall plots, this is sort of the best change in tumor size, plotted, where up means the tumor continued to grow in therapy, and down means the tumor shrank with therapy. And if you remember at the beginning, I introduced the response rate of partial response being about 8% overall in the last 62 studies. Well, at our recommended phase 2 dose, 3 out of 5 patients responded, so a PR of 60% or so. And I highlighted those patients all in with the asterisks above those individual responses. Now, we did have patients whose tumors shrank, but then had new lesions, and that's the progressive disease with tumor shrinkage, and so that's something to pay attention to. And many of these responses were really quite dramatic, is what you'd predict for if you inhibit the dominant oncogene of the tumor. So here's a patient that, on the top, went to the University of Michigan in a wheelchair for cycle 1 with this rather large tumor, and post 2 cycles of trabectedin walked into clinic, was looking good, was really quite a dramatic change, and Rashmi said she didn't even recognize him. Here's another patient. This was our first responder. This was actually at CHOP, and I was like looking at this on Epic at my like kitchen table, and it was like, that's pretty cool. But anyway, this kid had a 6-centimeter parapancreatic tumor, and after 4 cycles, there was no detectable tumor, and this kid went on to get 19 cycles of therapy, and this was a kid who had relapsed like 4 or 5 times, and transplant and everything else. So really quite exciting. And here's a couple other patients. The one on the top was an NCI patient that Mary Frances shared with me, so I don't see her here, but that's why I put it up there. And then the last one is one of our more recent patients. But even patients that don't technically fall under tumor regression, there was quite a bit of benefit from the regimen as well. So here's a patient who technically doesn't meet RECIST criteria, but you can see there was quite a dramatic change in this tumor, like it sort of calcified and looked very different. And this patient's quality of life was quite high, and was rapidly progressing when they went on our study, and then was able to have Thanksgiving and Christmas, which is something as well. All right, so what have we learned so far? So we've seen striking tumor regressions. It's early days. It's only phase 1, and it's 3 out of still a handful of patients. But I think that the regressions are consistent with the idea that we've repressed EWS-Fly, and I'll show you some data to that in a minute. Most patients have benefited. Patients with widely metastatic bone disease, we saw a lot of improvement in pain and quality of life. Unfortunately, we didn't capture that. And I would argue, actually, we criticize a lot our cell line models, and are they necessarily predictive of response? I would argue that if you're rigorous in how you do your xenograft experiments, actually they are, and they definitely prove to be here. These tumors regressed in our models, and they regressed in our patients, okay? And I think that just, comes back to like, let the tumor establish, let it grow, treat it, effectively, and be rigorous. Again, our patients were heavily pretreated, and so this does ask the question of like, are we really going to roll out every new agent in this patient population? I don't know that we have much of a choice at this point, but it's certainly going to challenge and push us to, and maybe perhaps lose some good agents along the way. But anyway, the future of this is, we're now accruing in our phase 2. We have 18 patients for this, and we've already enrolled 10, so that's in about 6 months, which is really great. And that'll be the next step. And then there's been talk about rolling out the regimen either in first relapse in the COG or as the next arm in recur. All right, so at the end of the day, and keeping with the cycle of our work, tumors regressed, but they recurred. So even our patient that got 19 cycles, he had a progressive lesion that actually we put in a PDX, and we're hopeful that will grow, which will be a really valuable resource. So what do we need? Well, we definitely need less toxicity. Like, we did see a fair amount of toxicity, and then we need to figure out how to extend the response. And so to do that, I think we've sort of going back to the bench and taking a couple different approaches. I'll talk a little bit about the correlative biology, which is going to guide some of those studies, but also I do think we can do quite a bit to find better combinations, with the idea that if we focus on the mechanisms that we've characterized, then maybe we can have more on-target combinations. Irinotecan is still a DNA-damaging agent. It's still a relatively non-specific potentiator of transcriptional repression. But in terms of a less toxic alternative, there is already a molecule that's in the clinic and has been evaluated in Ewing's with actually was a positive study, and that is this drug called lurbinectedin. And lurbinectedin is a second-generation trabectedin inhibitor second-generation trabectedin analog that accumulates to far higher serum levels. So if Cmax is the name of the game, this is like 20 times higher than what you need to inhibit EWS-Fly. And we published on this drug in 2016 and showed that it worked similar to trabectedin, and actually maybe a little bit more cleaner in terms of its repression of EWS-Fly. And so, this has allowed us to explore issues, again, getting to the fact of optimizing schedule administration of drug, is maybe we can use this compound, instead of just inhibiting EWS-Fly, maybe as a single agent we could sustain the response, because we're so far beyond what we need for Cmax, maybe we can divide the dose and extend the duration of EWS-Fly suppression. And Jeff Tretzky showed that in his work as well, that this is a different class of molecules. This is not just hit the target and run away. This is hit and extend, and then you'll see a more profound response. And so we modeled this in the mouse These drugs are really hard to work with in the mouse, and I just happen to have the best animal technician on the East Coast, with the possible exception of Arnulfo, for those of you who know Arnulfo. But she's amazing, and so like if you miss the tail vein in this, you lose the mouse. And so she was able to dose the mouse daily with lurbinectedin in a cohort of mice, and you see the data on the right. And this has really been transformative data for getting this study off of the ground. But again, these are spaghetti plots. On the left is vehicle. You can see the tumors grow quite aggressively with continuous suppression over just that narrow window of 5 consecutive doses. You can see sustained tumor regressions in every mouse in the cohort. Okay, so that's the hypothesis of our study that's going to open soon, that we can use this drug in a divided fashion to inhibit and sustain suppression of EWS fly. We also published along the way that this drug inhibits DSRCT by an analogous mechanism or EWSWT1 and DSRCT by an analogous mechanism that I presented for EWS fly. And so we've written the study as a phase 1 to find this divided dose schedule, and then a phase 2 in Ewing's, because that's where the majority of the data is, with an expansion cohort to include subsets of DSRCT patients. And this is the LIFT study. It's sponsored by Stand Up to Cancer. Again, it's many of my friends and people who it's really exciting to have interested in this biology and these compounds, including Ted Leitch, many of you people know from the infantile fibrosarcoma. He co-leads this at CHOP. CHOP's actually sponsoring the study. And Bob Mackey, who was at Penn at the time but moved to Memorial. And then again, my friends at the NCI, John Glad and Mary Francis, and many of the same people from the SARC-37 study will be on it. And much of the correlative biology will be done at the NCI as well. And then we also recruited Julia Gladebender, who is one of the world leaders, sarcoma clinical trialists. But anyway, we recently just, the idea is we were going to get through our IRB at our institution and then expand to the other sites. And so we just recently were approved to open. So we're open. We're hopefully going to enroll our first patient. Again, it'll be an all-FET fusion. So some of these rare fusion types like EWSR1 and FATC2, that we all know it's a bad tumor, but like, what do you do with that patient? As well as EWSWT1 and FUSE-ERG and all these other rare fusions. Our first patient will be an EWS-ERG, which we excluded from SARC-37 because we just didn't know. All right, so in terms of the continuous process between bench and bedside and back, one of the things that I think is pivotal to these studies is when you let the biologist help guide the study, you do a lot of correlative biology, which for a coordinator is kind of a nightmare because we're shipping tumors and tissue all over the study and circulating tumor DNA, and so, you name it, it's going. I have a map where it shows all the arrows of where everything goes, and it's kind of a nightmare, but really I think it's absolutely just as important as the results of the study, because unless we really understand what's different about these patients, we're not going to make progress with these agents or with any other approach, I would argue. So this is, some of the example of the correlative biology that we did on SARC-37. All the tumors were genetically profiled, so everyone got sort of a standard Archer solid tumor panel. We had this 18F-FLT biomarker, which we had published on, reflects EWS fly activity, that hasn't really panned out so far, but it's early days. And we collected circulating tumor cells and circulating tumor DNA that were really going to be, there's going to be a lot of information garnered from those. And these are the individuals that sort of, and we established PDX models in a majority of these or in several of these patients. So we will have at the end of this study trabectedin resistant, trabectedin sensitive, and trabectedin intermediate PDX models to help guide the future studies. And again, it only comes with really great collaborations like Ryan Roberts at Nationwide coming in on Saturday to put a PDX model in a mouse. Steve Du Bois and Brian Crompton at Dana-Farber with circulating tumor DNA, and Mas Hayashi, another Hopkins, one of us, Hopkins NCI person. And so in the last couple of minutes, I'm just going to, last 15 minutes or so, talk about how we think the EWS fly transcriptome is in many ways going to guide us on the way forward, and how we collected in some patients pre and post treatment RNA sequencing and pre and post viably frozen cells that we'll be able to do single cell RNA sequencing to really understand sort of the EWS fly transcriptome in patients. All right, so this is data I've never presented before of a patient on our study who had a partial response. It was one of the one of our better responses actually, that we got biopsies pre and post-treatment. And this is actually RNA sequencing from FFPE, not frozen samples. And Leah Suri, shown there on the bottom right, is a molecular pathologist at CHOP who's really amazing, and Rebecca Kaufman is our bioinformatics scientist who's analyzed this data. But this patient had, again, a nice response, and we were able to do RNA sequencing before the drug and then about 16 to 18 hours after drug treatment. And what I'm showing you is just a basic heat map of expression, and on the top are targets induced by EWS fly, and on the bottom are sort of highlighted by the teal are targets that are suppressed by EWS fly. And you can see with drug treatment, really, rather quite remarkably, I didn't think this was actually going to work. The red becomes blue, and the blue becomes red, indicative of flipping the EWS fly transcriptome in patients in the clinic. Now, what was quite interesting about it was this is another patient from the same RNA sequencing run where we did the same thing pre and post. These are the exact same targets, and they line up, so I cropped it just for ease of view. But you notice in this patient also had a response, but their tumor went less red to blue and less blue to red. And also, if you look sort of at baseline pre, the transcriptional profiles are very different, actually, right? So if you look at, try to compare one line to the next, even if you just look at the top few on the left, those are really red, and on the right, they're not even expressed. They're, in fact, probably almost look like suppressed genes, suggesting that, there's quite a bit of difference between these two patients in the transcriptome, and or there's sampling bias based on where we put the needle to get our tiny little core to do the sequencing, or there's, a lot of other potential explanations. But to me, it suggested that there's quite a bit of difference in the transcriptome from patient to patient, and there was a reason why we thought that. Okay, so the heterogeneity in the transcriptome is obvious. Is this an assay artifact? Is there something about sampling bias or RNA sequencing, or is transcriptional heterogeneity a fundamental feature of the EWS fly transcriptome? And this is actually a question we asked years ago, just looking at our RNA sequencing data, and Jeff and I were actually talking about it last night, that, it flipped it, but there was, it wasn't perfect. And so one thing we excluded was maybe it was just contaminated with fibroblasts or whatever, and that was the difference. And as you can see here, I'm not going to really line up behind this data because it's still pretty preliminary, but for the most part, the vast majority of the sequencing reads were pure tumor and not infiltrating lymphocytes, sorry, immunologists, or contaminating fibroblasts or something. Okay, so again, a few years ago we set out to define what is the EWS fly transcriptome and what exactly are the targets, and there was a number of publications which I summarize here where they just sort of characterize the gene signature of EWS fly, and in general everybody sort of knocks it down and looks at some log fold change threshold across tumor types, plus or minus some chip data or some patient sample, and arrive at sort of similar genes. You can see targets and go, oh, that's an EWS fly target. But certainly there was enough difference among all of these signatures to suggest that that transcriptional heterogeneity might be a fundamental feature of the disease. And if you look at sort of Steve Lesnick took this on over a decade ago and looked at all published gene signatures and did a meta-analysis to arrive at sort of the putative set of targets. And if you look at the data from that paper, this graph just shows the number of lists a given target appeared on. So for all the sequencing that people have done, how many times did this particular target recur? And you can see the top target only appeared on 60% of the lists. And well-established targets like NR0B1 was even less than that. And it was even worse for repressed targets. So the top target or repressed target only appeared on 35% of the lists. So again, is this heterogeneity an assay artifact? These were different techniques, there were different labs, there were different cell lines, or is it actually a fundamental feature of the transcriptome? And so we worked to really optimize the knockdown and make it quite consistent across 6 different cell lines. This took a lot of time, and it was utterly painful. We optimized lipid concentration, cell density, time of incubation, with the idea that we would sequence at the earliest point following suppression, such that we limited indirect targets, but really made consistent suppression across multiple cell lines, and all those factors mattered. Okay, and so then we did the sequencing and we collect the RNA, and this is just looking at did we achieve relatively comparable knockdown of EWS fly across cell lines? And so this is an absolute quantitation, so quantitation of the expression level versus a standard curve, so we know the exact sort of nanogram amount of EWS fly. And one of the things you notice right away is if you just sort of look at all the dark gray boxes, there's quite a bit of a different level in expression from model to model, which, I guess makes sense kind of, but we've known for a lot of years that EWS fly doesn't tolerate changes in expression one way or the other. If you increase it, it's just as bad on the cells as if you decrease it. And so it's quite interesting that the different cell lines find a different steady state. But I would argue that we had relatively comparable suppression as indicated by the lowest bars to the right across all these different 6 cell lines. Okay, and so we asked the question, is there heterogeneity? And so first thing we did is just sort of look at our gene expression changes as a function of the published gene sets. Okay, so these are just old school heat maps looking at log-fold change between control and treated, where blue is down and red is up, segregated by induced and repressed targets. The labeling is a little off in the middle, but you can see that, it looks like the group of induced targets turn blue and the group of repressed targets turn red, and so that would be consistent with silencing. But again, if you drill down and look at individual rows, it's not at all the same. Like some that doesn't change, some exactly the same, some go up, some go down, and in fact we saw many genes that flipped in the opposite direction. And so if you actually look bioinformatically at is this statistically significant suppression? So if you look at these gene sets, there's a total of 9 gene sets. So 4 sets of induced targets, 4 sets of repressed targets, and then one that was sort of a hodgepodge. Only about half of them end up being statistically significant with direct silencing of the fusion protein. So it's kind of a terrible assay if you think about it, right? You're looking at a gene set that's supposed to repress EWS or reflect EWS fly silencing. You're looking at data generated by silencing EWS fly that you confirmed rather robustly that you did silence the target, and it's not significant in almost half of the cases. And so the number of significant gene sets differed by cell line. So in A673, 5 of the 9 cell lines were significant. TC71 was 0, and TC32 was only a third were significant. And same with the data sets. Total of 12 cell lines. If you look at it by cell line, so there's 12 cell lines, there's 6 induced and 6 repressed sets. Again, like a third to 25% are actually significant using real statistics in bioinformatics, and that's graphed on the bottom there. So what does this mean for what genes are common, what genes are different? So I think we can draw some conclusions about if you just look at the protein coding genes and set a threshold cutoff of 0.001, you can see that if, in general, if you look across common targets, there's about 22,000 of those that are common across all the cell lines. So why are they not more significant then? Well, I'll talk about that in a minute, but it depends on magnitude. But there's easily at least as many cell line unique targets with this statistical cutoff. So there's 728 in just TC32s, or 652 unique significant gene expression changes in an individual cell line. So the approach that people commonly use then to decide on what are real gene expression changes aside from statistics is to set a threshold. So what if you set a relatively rigorous threshold and say, all right, well, a log-fold change of 2, so a four-fold change with a p-value of 0.01, how many of those are common across all 6 cell lines? There's only 5, actually, in our data set, with pretty rigorous silencing and similar changes, right? Instead, what you see is these large magnitude gene expression changes tend to be cell line unique. And so there's 670 unique gene expression changes in, for example, A673 cells that are totally unique to that, but are a log-fold 2 change and are significant. And if you push it even farther, drop the p-value to 1 × 10 to the negative 5th and say it's got to be almost an order of magnitude change difference, there's 150 genes across in this case, across all 6 cell lines that change by a log-fold 2 change of 3 or more. So really quite a stunning number. And many of these, if you look at the heat map, change by log 2-fold change of 10. So that's almost a 1,000-fold change in expression. So if you're thinking about the biology of an EWS fly-driven gene, what's more important, something that changes by a small magnitude across all the models, or something that goes up by 1,000-fold in just one model? And I don't know the answer to that, but I'm pretty sure that it's important. And so then, like what's quite the obvious next question is, like, how does this heterogeneity exist across cells within a model? Because again, we have now patient data with, or viably frozen patient tumors that we can look at the heterogeneity as a function of drug treatment in patients in the clinic. And so, these are UMAP plots, so they're also designed to find differences, so it might be a little bit of a biased data set, and it's really early days with these data. I'm just sort of presenting it here in the last couple minutes to sort of get people thinking a little bit. But, if you look at the top, we took sort of 4 of our cell lines with the optimized conditions, and we silenced EWS fly and looked at the transcriptional profile, and those are the UMAP plots where the media are sort of salmon colored, and the EWS fly definitely leads to a shift on the UMAP plot to the left. But if you look at just sort of the blue sections and then compare across cell lines, it's quite dramatic how these different clusters are represented to different degrees in different cell lines. So consistent with this idea that there's transcriptional heterogeneity among lines, there's also heterogeneity within a given tumor in subsets of cells. So it's really quite a staggering difference. So if you look at like the cluster 5 genes, the sort of teal ones, like they're almost absent in CHLA9, and TC32, they're quite profound, and perhaps even more stunning is, CHLA9 and CHLA10 come from the same patient at diagnosis and relapse, and you can see there's quite a difference in the transcriptional program with silent in the EWS fly target genes, actually, because that's the silenced cluster between these 2 samples taken serially from the same patient. And so, the question is this a mechanism of selection? And are we watching sort of the evolution of a gene expression program that maybe eventually overtakes the whole tumor? I don't know. That's going to take years to figure it out. But I would suggest that, some of the common targets, if you look at the UMAP plots on the top for NR-OB1, there's induced targets to the right, and then it sort of suppresses to the left, but that's also not really uniform. There's some cells that maybe don't see suppression, even in tissue culture. Maybe that's also an assay artifact, I'm not sure. But if you sort of drill down on some of these unique gene expression changes that were large magnitude, you can sort of see in the CHLA10 cells sort of this pocket of really highly expressed of that particular target. I don't even know what it is, but it does suggest sort of an evolution that may be in fact mediated by EWS fly. And again, you can sort of define these clusters by these large magnitude gene expression changes. These were just sort of the top 5 largest magnitude gene expression changes in each of the clusters. And Jeff's looking at this list and he's like, EWS fly target, EWS fly target, EWS fly target. So many of them are EWS fly targets. All right, so I think just to sort of summarize what I've talked about, Ewing sarcoma is definitely dependent on EWS fly, and it is indeed a druggable target. SARC37 has been really an exciting study, and it's been really quite fulfilling to see these dramatic responses in these patients, and more importantly, the improvement in quality of life for many of them. And we actually have now evidence that we did in fact hit the target in patients in the clinic, which to my knowledge has not been shown before. And I didn't think that assay would actually work because of the heterogeneity. But unfortunately, tumors have regrown and progressed, and so we're working on identifying analogs with better toxicity profiles, better combinations. We just were funded with an R1 to look at sort of mechanistically focused combinations and then also to try to understand this evolution of transcriptional programming. And importantly, we've established models that we know are resistant to our drugs, which should be really cool. We can do the actual experiment where you don't even have to biopsy. You can do a whole tumor if you need to in an animal and treat with the drug combination. I do think I'll make the case, and I'll be interested to hear feedback from some of the Ewing's experts in the room, that heterogeneity, I do believe, is a fundamental feature of the transcriptome, and I think we've shown that here. And if you sort of think about the biology of transcription and the biology of EWS fly, where it's sort of like this transcription factory that slides chromatin through, like it would make sense that this nidus of transcriptional badness could sort of select for what it needs. And so I think that this is really an important question that we're looking at both in the single cell level and chromatin-wise, and then also using cut and tag and some of these newer EWS fly mapping techniques. All right, so this is my lab. Nothing's possible without them, and it's really a great place to work. So if people are interested, let me know. But Alyssa Levine is the one I told you about. She's right in front of me there, and she's our animal technician. And then Seneca to her left was a postbac who did all this wonderful work with some of this analysis of tumor heterogeneity. And Rebecca Kaufman is our fearless bioinformatician who I'm like, we're going to do GrowSeq, we're going to do cut and tag, and she just like charges after it, so it's really awesome. And I do also want to acknowledge so many great collaborators that we have, and I want to acknowledge everybody in this room because I just really love this community and this environment, and the NCI has really supported these studies, and it's really helpful when you're sort of piecing together grants to try to get a clinical trial done to have the NCI invest in the study as well. And so I thank Brigitte for that. She's lined up behind these studies, and then again, John and Mary Frances have been great. And then this is our funding, and those are our buildings. And thank you for your attention. I'm happy to take any questions.
Do we have microphones going around? I'll give you one. Oh, okay. There's one coming too.
Thank you for that. It's always so gratifying to see people's hard work at the bench result in actual benefits for patients. I was wondering if you or others have attempted to narrow down that list of heterogeneous EWS-FLI1 targets using either targeted CRISPR screening or quantitative proteomics. There's a lot of variability in the transcriptome, as you point out, and it doesn't always get reflected in the proteome. And so that may be a way to kind of filter down those.
Yeah, Thomas Grunwald in Germany has done a fair bit of proteomics work looking at sort of differences in the transcriptome and the proteome, and I absolutely agree that that's important. And we've started looking at some of those mechanisms in terms of like what's the translation of EWS-FLI require, and additionally downstream. And as you know, it's pretty complicated, and but I think it's a really good idea. And as far as the screens, we've done a lot of screens, but mostly looking at sort of what potentiates EWS-FLI, either expression or activity. But yeah, certainly like targeted persister screens or whatever would be really cool to do as well.
I thought this was a really intriguing, intriguing talk. Thank you so much. I was wondering, I was intrigued by your slide with the CHLA 9 and 10 data set, because it looked like that although it was pre and post treatment, there were some clusters that were still similar and populated by both plots. So I guess my question for you, looking at tumor heterogeneity and clonal evolution, do you think, let's say you treat, the tumor burden shrinks down, but as the tumor evolves again, that there is an imprinted kind of epigenome that predisposes that signature of transcriptome to reoccur? And what are your thoughts on potential factors that could imprint the epigenome in this regard?
Yeah, I mean, I think that's absolutely the critical question: is what is the long-term effect on the epigenome, and what is the long-term effect on the epigenome by targeting EWS-FLI? And actually, my guess is that this is purely a guess, again, sort of informed by clinical responses, is that it's an epigenome reset. That by targeting EWS-FLI, you can sort of reset the epigenome. And the only real evidence I have for that is, we've noticed that some of our patients have come off our studies, have gone on to other studies, and have done pretty well after our study. So like they might have just selected the best next study, but it is an intriguing idea that it's not imprinted, and that you can do sort of an epigenome reset by targeting the fusion protein. I think either way, it's a therapeutic vulnerability, right? If it's imprinted, then you probably need to develop approaches to trap that. If it resets, then it sort of gives you a fresh shot on goal with a newer tumor. But yeah, I think if you can understand that relationship, that's really the critical question. Yeah, so I mean, all of this does have attack sequencing. We just haven't really quite gotten to that data yet. So we will be able to answer the question about what's the change in chromatin structure. And actually what's going to be super cool is Brian Crompton is our collaborator, and he's looking at trying to infer chromatin structure from the circulating free tumor DNA in patient samples. And so that might be an opportunity to sort of see how that's evolving in the tumor and draw some conclusions. But yeah, I mean, it's probably like a decade and a half of work.
Jeff.
So, brilliant summary of a lot of work, and really to be congratulated on following a lot of these things along and getting to the point of doing this. One point I just want to make with regard to the last speaker, or last question, was that, we've actually known for a long time that Ewing sarcoma tumors do respond to retreatments. You can retreat a Ewing sarcoma even with the same drugs after they recur, and they will respond for a short period of time. In fact, when I was a fellow at the NCI, we were using this regimen of low-dose chemotherapy called EPOC, and patients who would relapse after getting big doses of the standard drugs would get those essentially the same drugs as a low-dose continuous infusion for 4 days, and they had really good responses. So there's something about this tumor, and I don't know whether we can pin it on epigenetics or whether it's clonal regrowth of the sensitive cells, but I think there's something interesting there, and it begs the question ultimately of, why are we having so much trouble getting rid of it? Which leads me to my question, which is, so congratulations on putting that Stand Up to Cancer LIFT trial together. I'm very excited to see how that's going to go. But I'm also curious on how you're putting the mechanism of targeting all of the FET proteins together with LIRB and why you think that whole group of fusion proteins are going to respond, or well, you've seen responses, but what mechanistically holds them all together?
So you remember the original rationale was that FUSE-CHOP was the original first winner, so FUSE and EWSR1 being family members. And then we published a paper in DSRCT where we showed that in that tumor type, EWSWT1 relocalizes in a similar way as EWS-FLI, and it flips the transcriptome. So based on those 3 tumor types, we don't know about FUSE-CHOP. We would argue that it might be all FET tumors. I would love to look preclinically before opening that study, but as you know, there's no models of EWSR1 and FAT-C2 or some of these rare fusions. So I think that the only way you're going to be able to ask and answer that question is in patients. In terms of like PD markers, we're going to look actually, can we see relocalization of the fusion proteins in patient samples pre and post treatment and FFPE samples. So that'll tell us whether that correlates. And then really the final piece will be nailing down exactly what's driving the relocalization, what are the pathways involved, and that sort of things, because we actually don't have a total understanding of that. We have some data on it, but not a complete understanding. But to your first point though about, for the trainees in the room, and Jeff, you're an example of this as well. If you have really good data and you really believe in it, you have to stay with it, right? You can't. We took a lot of heat for, that drug failed. Why are you still working on it, right? But if you believe in what you do, then you really need to follow it through.
That was really nice work. As far as the mechanism of action for these drugs inhibiting these different transcription factor fusions. It's been known that different chemotherapy agents, they don't just go into the cell, but they actually compartmentalize into different regions of the cell. So do you have any evidence that these drugs actually go into EWS-FLI nuclear condensates specifically?
We do not, no. We've talked to some of the nuclear condensate people to look at that. I think the hard part of this approach when you start in the clinic and you have a complicated molecule like trabectedin is the mechanisms are really complicated, right? But one thing I think that we've learned is as we peel back layers, every time we peel back a layer and learn more, this might be another example, then it reveals a vulnerability. And so I would say we are going to map binding genomically. Those studies are underway. And I would predict that the actual event, the DNA binding and damage, happens at EWS-FLI response elements. That was one of the original theories as well, based on published preferred binding of this class of drugs. And then that opens up a whole suite of new studies and new combinations. But thank you for the question.
Natasha.
2 questions. Fascinating talk, great work, and I love your persistence on something that a lot of people were like, this is never going to work, so bravo to you. My first question is about drug penetration, or access, I guess, to the tumor. Is anything you can say from the PET studies or from your single cell that suggests that's playing a role in some way?
Yeah, so you're absolutely right. The distribution of suppression in a tumor is not at all uniform, and we published that a couple years ago. So we used this PET tracer to reflect EWS fly activity, because EWS fly sort of drives the nuclear transport proteins that bring the tracer into the cell. And so we reasoned a few years ago in our preclinical model that you could sort of rebuild the tumor in 3 dimensions and make like a heat map of target suppression in sort of a crude way. And suppression was not at all uniform in even, like, this is genetically identical mice with a cell line xenograft from the same tube and the drug coming from the same tube on the same day. Like, everything was, and again, I have the best technician other than Arnulfo in the planet. And from xenograft to xenograft, the suppression was vastly different. And so we started to collaborate with a woman named Carrie Shadler at MD Anderson Cancer Center, who's a vascular biologist who's looked at issues related to drug delivery and permeability. And what she's shown is that when drugs hit the tumor margin, they like completely go sideways. So they around the periphery of the tumor, these vessels are totally leaky, and the drugs sort of pour out. And she can show that by doing DAPI stainings. And if she exercises the mouse, she can rescue some of that behavior and lead to more effective delivery. So I absolutely think that that's an important mechanism that needs to be teased apart. We wrote some grants on it, didn't get them, so it kind of sits on the shelf.
Yeah, I mean, my second question is related to that, because I think these are stromal cells, the cell of origin of Ewing's. It's probably some mesenchymal cell. I don't know, maybe neuro mesenchymal hybrid thing, and they make ECM. And I wondered if you looked at ECM in that way, and I think the vasculature relates to the ECM. So,
yeah, we've taken some looks at the secretome to the point that was made earlier about the protein targets, and like for sure, there's plenty of candidates for why that's happening. But I think that'll be an important. That's even just in cell culture with silencing EWS fly. You can see plenty of candidates that would cause that, but like that doesn't even involve the level of complexity you're talking about, like. What is EWS fly driving, it's being secreted, that's activating, name the fibroblast or endothelial cell that's changing everything. So I think it's also a really important area of study. Good question.
Awesome.
So Patrick, I'll ask the last question then. You alluded that all these trials are failing, but maybe the drugs are not failing, the trials are failing. So are we not testing the drugs in the right way? Our expectations are too high. When we test a new drug, maybe shall we focus on to prove that the drug is actually doing at the molecular level, hitting the target, and if it is good enough now, go put it in a frontline treatment. So in order to change this whole clinical trial culture for testing new compounds, what are the barriers? Is it the ethical issue? Is it the regulatory issue? Or is it just the clinicians changing their mindset?
I think it's complicated, right? So, many of these drugs are proprietary, and, we're sort of begging for them for pediatrics to begin with. And so, for companies to try a new dose or a new schedule takes quite a bit of work. But I do wonder, of those 5 negative phase 2s, if we spent like a year working on issues related to target suppression, like, think about like all the VEGF inhibitors, like, we just sort of gave it to the patients and didn't work and we were done. But like, did we really look at like, well, how long do we need to expose the cell? Like, we tend to go with a 21-day cycle. Is that really necessary? I'm not sure. And, I'll just give you one other example. We have a new paper that we're submitting soon on a, more chemo-like combination that when we tested the published schedule and the schedule that's been used in multiple clinical trials, it didn't work at all. And then when we optimized it, like the tumors went like that. So I think there's going to be a lot of evidence from that, and I think that this is a way we should leverage our preclinical models and these huge PDX collections that we're building up and really go after that question. But I mean, your work and Jeff's work looking at that very carefully with the TK compound was, is exactly emblematic of that really matters.
Thank you very much.
Thank you, everybody.
Yeah, the other thing I just want to add to the question of why these drug trials haven't worked, Patrick, is those weren't drugs that were ever designed for those cancers. I mean, a lot of those drugs in those phase 2 trials were designed for other cancers, and they were drug companies saying, well, if we can get a pediatric indication, so it goes into a phase 2 trial in pediatrics, and I think that's a huge challenge that we all need to face, which is that there are targets for pediatric cancer that drug companies aren't going after. We need to go after them, like, what we're doing. And the fact is, if you take, a bunch of drugs designed for breast cancer and then throw it into a Ewing sarcoma model or a Ewing sarcoma patient, it may not be very effective. And it may not be very effective because it's the wrong drug, because that target is not a critical target in that disease. And that's really important too. So the combination of pharmacology and the wrong target, wrong disease are at least two of many reasons why, so we've got some work to do for targets. Speaking of targets, we have many wonderful posters with lots of great ideas, targets, and enthusiastic presenters that are going to share this with you. The way we're going to do things now, we're going to break for lunch.